Hi, everyone. Welcome to OsteoBytes. Happy Thursday. Thank you for joining us. We will give a few seconds for everyone to get situated in our virtual room. I'm so glad that you took the time to join us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon. I am Director of Scientific Programs at MIB Agents. And today on OsteoBytes, we are talking with Dr. Ming Li from Memorial Sloan Kettering Cancer Center. Dr. Li will be discussing discrete classes of tumor-elicited immunity and tolerance responses, which support cancer immunology as a new discipline that transcends the classical self versus non-self doctrine, with the underlying molecular and cellular circuitries exploitable for cancer therapies. Thank you so much, Dr. Li, for joining us on OsteoBytes today. We're thrilled to have you. And thank you also to Walker, our panelist, for joining us. A little bit more about our guest today. Dr. Li is a member of the Immunology Program at Memorial Sloan Kettering Cancer Center and professor at the Weill Cornell Graduate School of Medical Sciences. His laboratory studies immune regulation and its relevance to diseases, including cancer, and recent work has focused on the signaling, metabolic, and transcriptional mechanisms of T-cell regulation, as well as tumor-elicited innate and adaptive immune responses, and how this knowledge can be translated for novel cancer immunotherapy. Dr. Li received his PhD in biological sciences from Columbia University and postdoctoral training at Yale University. Dr. Li is an American Academy of Arts and Sciences Fellow and has received a number of honors, including a Howard Hughes Medical Institute Faculty Scholar Award, a Rita Allen Foundation Scholar Award, an American Cancer Society Scholar Award, and the 2016 American Association of Immunologists Bioscience Investigator Award for outstanding contributions to the field of immunology. So your fireplace mantle must be like super full, Dr. Li, with all these awards. You're running out of room. Okay, some announcements and reminders before we get started. We have a couple of deadlines coming up for our Outsmarting Research Grant and for our Factor Conference abstracts. So our 2024 cycle of our Outsmarting Osteosarcoma Grant Program is now open, and you can find the application on Proposal Central. Applications are due January 26th, and you can check our website for more information on the program guidelines. You can see past recipients, and we also have a YI Research Mentor Program that's new this year. We have a short 15-minute video that you can watch to learn more about the application and review process. And if you have any questions, you can always email me at christina@mibagents.org, and I'll put a link in the chat with more info. And we're now also accepting abstract submissions for Factor 2024, which is our annual osteosarcoma conference. It's going to be in Cleveland, June 20th to 22nd this year. Abstracts are due January 12th. And that application is also available on Proposal Central. And I'll also put more info in the chat about that. And then we just launched a Google group for medical professionals with a clinical or research interest in osteosarcoma. It's called the Osteosarcoma Medical Discussion Network, which is a mouthful. So we can just call it OSMDNet for short. It's a closed, moderated group, and you can request to join on our website. And I'll also put a link to that in the chat. And lastly, for our bereaved families, we have 2 Healing Heart Grief coaching sessions for bereaved parents this month. There's a meeting on Wednesday, December 6th at 7 Eastern. And we also have a special session on getting through the holidays. You know, this is a difficult time of year. On Wednesday, December 13th at 7 Eastern as well. And to RSVP or get more information, you can email isabel@mibagents.org. And I'll also put some more info in the chat about that. All right, Walker, can you please introduce yourself?
Yeah, hi, my name's Walker. I'm an osteo warrior. I'm this year's MIB Junior Advisory Board Vice President, and I'm very excited to hear what Dr. Li has to say today. So from there, you can take it. Can't wait to hear what you have to say.
Thank you so much, Christina, for the invitation and the introduction of Walker for monitoring the session. Let me just share my screen. Can you all see the slides?
Yes, that looks great.
As Christina introduced, so one of my lab's interests is really to understand how the immune system senses response to cancer, and in this context, how the immune system can be targeted for effective cancer therapy. Now, I would just start by sort of giving some background in terms of how we perceive cancer as a distinct disease. So a lot of emphasis has been on essentially how cancer cell can acquire mutations and thus gain this growth advantage. But I think in recent years, it started to be appreciated that clonally expanded cancer cells have ultimately engaged a supportive environment, and to the point that the sort of destruction of local tissue environment that result in abnormal invasion growth is more of a defining feature of cancer as a disease. So this more adaptation tissue environment will involve, to a great extent, the engagement of aberrant blood vasculature, or so-called angiogenesis, as the process of one of the cancer hallmarks. Now, in this context, if we think about cancer therapy, we may not necessarily have to be eliminate all these clonally expanded cells, as long as we could reverse this sort of more adapted environment to one that is not as much supportive for abnormal cancer cell growth, that could still also be a viable cancer therapy approach, as has been shown for various anti-angiogenic type of therapeutic drugs. Now, as you probably start to appreciate that one of the important populations of cells in this dysregulated tissue environment are these various type of cells of the hematopoietic cell lineage, or immune cells. That's essentially the interest of the general cancer immunology field and also my laboratory. Essentially, we want to understand the diverse types of immune cells that would respond to a growing tumor, and understand the principles that guide the infiltration of these cells, and how possibly these cells can be targeted for cancer therapy. So that's the topic I want to share with you today. Now, for immunologists, we wish to differentiate different classes of immune responses. So in this case, we wish to differentiate more well-studied adaptive immune response to cancer, or what we call primary dependent immunity. Because for this class of adaptive T cell response, the lymphocyte, in this case, T lymphocyte, B lymphocytes, they have to see antigen signal lymphoid organs before they can possibly migrate to the tumor tissue. Now, there's another class of immune response, so-called pre-wild immunity. So this will involve other types of lymphoid cells, such as innate lymphocytes, but as well as more innate-like T cells, and also myeloid cells, such as macrophage. So these cells, following differentiation in primary lymphoid organ, such as the bone marrow or thymus, they can directly migrate to the tissue and sense the local environmental change. So I think it's important sometimes to really broadly differentiate different class immune response that we could characterize. Now, in this context, the advance in the cancer immunology field has been on this primary dependent immunity arm. It is because of following decades of studies which to review, for instance, T lymphocytes, their activity can be tuned by both positive signals and also negative signals. And these negative signals, so-called immune checkpoint inhibitor pathways, they can be targeted to revive anti-tumor immunity. As probably all of you know, this have really transformed cancer therapy in general. This just listed a few classical studies in this particular field. Now, so my lab is also interested in adaptive T cell response in cancer, and see how whether the additional pathway may be targeted for cancer therapy. In fact, one of the pathways we studied is a TGL-beta signaling pathway. We could show TGL-beta can be also a really critical negative regulator of adaptive immunity, or specifically CD4 T cell mediated adaptive immunity. But in terms of therapeutic targeting, TGL-beta is much more complex pathway than PD-1 or CTLA-4 pathway, to the point we have engineered a more targeted blocking approach. Essentially, we have an anti-CD4 directed TGL-beta inhibitor, so that we can have more of a selective target effect in terms of promoting anti-tumor immunity. So that's sort of the topic I don't have time to share with you today in detail, but that's also obviously we are interested in. Now, in addition to this small so-called primary dependent cancer immunity, we're also interested, our lab, in fact, was the first to identify these alternative lineages of lymphocytes and their role in cancer immunity. So this will include more these innate lymphoid cells, or in short, the type 1 innate lymphoid cell, IC1s, as well as these killer innate like T cells. These are the class of lymphocytes most sense stress molecules instead of antigens are the major driver of their response in the tissue. So we're also interested in targeting this class of immune response for cancer therapy. But for today's discussion, we might talk about this other type of tissue-resident immune cell population called macrophages. Now, macrophages are considered present in almost all tissues, and broadly can define based on their location in the tissue. They can be macrophages associated with mobile parenchyma cells of specific tissue. Parenchyma cells define the cell that carry a tissue function, rather than more supporting stromal cell population, which can also have the macrophage residence. This is what typically called interstitial macrophage subsets. Now, in a growing tumor, typically there's a transformation of the parenchyma cells that drive, in this case, let's say a carcinoma in various tissues. So we could also have different classes of macrophages. The macrophage associated with expanding parenchyma tissue, which we call parenchyma TAMs, they can have one different kind of differentiation phenotype, and will be also different from this mobile altered interstitial macrophages that reside in the tumor microenvironment. Now, with this sort of broad differentiation, different macrophage subsets, I will just go to the model we have used quite extensively in the lab to understand these broad classes of immune responses in cancer. The model we have used so far involving a transgenic model breast cancer driven by this polyamido T oncoprotein that activate the canonical oncogenic pathways such as PI3 kinase and MAP kinase pathways. Recapitulates more this luminal type of breast cancer, and in fact, this also associated with expression of biomarkers associated with human cancer poor prognosis. So with this particular model years ago, we defined these two different macrophage populations defined by differential expression of these markers, for instance, CD11B. So the one of this macrophage population very much abundant in the healthy non-transformed mammary gland has high expression of CD11B, which we call mammary tissue macrophage, is phenotypically different from this tumor-associated macrophage, really expanded with progression of the tumor in this polyamido T model. So these 11B low ones, we named it tumor-associated macrophages. So with that story, we further show these macrophage differentiate from monocytes. They can have other differentiation markers such as TAMs express high levels of VCAM1, MTM express CD206, as well as a TAMs differentiation can be driven by a unique signaling pathway, such as a notch signaling pathway. So we are curious to know these different phenotypically different macrophages might have different tissue localization in the tumor microenvironment. So this end, we use these markers, essentially VCAM1 and 206 respectively, to define TAMs and MTMs. Which really stood out was that essentially the tumor-associated macrophage, they are indeed parenchyma associated. They are really nested in an E-cadherin positive cancer cell nest. And this is in contrast to this 206 positive cells, is more can be classified as interstitial macrophage population. These are the sort of MTM population. Now, with this unique localization TAMs in the tumor parenchyma, we have the follow-up study, in fact, published last year to show the tumor-associated macrophages have immune regulation function. So in this case, regulation CD8 T cell response to the point these tumor-associated macrophages can cross-present antigen to ultimately drive the CD8 T cell dysfunction following the activation by dendritic cells, another type of myeloid cells that can promote more CD8 T cell response. So that's associated with expansion, accumulation of so-called exhausted dysfunctional CD8 T cell in the tumor microenvironment. But that's again, this is a story published last year. But we are also curious to understand further differential regulation of tumor-associated macrophages and also these mammary tissue macrophages, again, defined by these different markers. So one observation we made, we looked closely into these two different subsets of macrophages is that tumor-associated macrophages appear to have really a smaller cell size than this mammary tissue macrophage. And cell size is really regulated by this major metabolic signaling pathway called mTORC1. So mTORC1, in fact, is indeed more highly activated in mammary tissue macrophage than tumor-associated macrophages. Is sort of based on this phospho-S6 staining, which is the mTORC1 signaling marker, as sort of quantified here. And so, mTORC1 is known to integrate various growth factor, nutrient, other microenvironment, as well as intracellular signaling, such as energetic state of the cell, to really collectively control this metabolic switch. So in terms of regulation and mTORC1 activation, there are two critical regulators, one around this TSC family of GTP accelerating protein, which is sort of a gap for this RIP small GTPase. So RIP is a small GTPase that can activate mTORC1. And TSC complex is a suppressive negative regulator of this pathway. So TSC really integrates multiple upstream signaling pathway in terms of growth factor and also energetic state through MP kinase in terms of sensing the signals to control mTORC1 signaling. So mTORC1 can also be regulated by the nutrient signaling pathway. The best defined is the RAC family small GTPase dependent nutrient sensing, which in fact will recruit TORC1 to the surface of lysosomes to be active by RIP small GTPase. So that's also will be a pathway I'm going to discuss with you for the second part of the presentation. So, considering TAM has low TORC1 activity in the PRMT model, we asked the question: could this low activity mediated by the TSC associated mechanisms? So, to this end, we deleted TSC1 with a conditional targeting approach. That indeed, in macrophage, in tumor-associated macrophages, that indeed resulted in enhanced mTORC1 signaling in VCAM1 positive tumor-associated macrophages. That is also associated with increased growth reflected by big cell size of these tumor-associated macrophages. So this reprogramming, in fact, is associated with quite a striking tumor suppression phenotype. The tumor growth is substantially inhibited. We further ruled out this is a response might be mediated by adaptive lymphocytes, because after we cross these mice to the RAC deficient background, which do not have adaptive lymphocytes, the protective phenotype still preserved. Then we wondered how could this reprogramming in terms of macrophage mTORC1 signaling might result in tumor suppression. So we took a closer look in terms of where these macrophages, TSC deficient macrophages, may have localized in the tumor tissue. They had this striking observation. So as I showed you previously, tumor-associated macrophage in control PRMT tumors, they really reside in the tumor bed, intermingled with E-cadherin positive cancer cells. But the TSC1 deficient tumor-associated macrophage now they really relocated more to the paravascular region, close to the vasculature. So this sort of quantification in terms of distance of the tumor-associated macrophage to the vicinity of the tumor-associated macrophage to the closest location of C31 positive endothelial cells. So in this case, you see the TSC deficient can result in the translocation of macrophage more in association with the C31 positive endothelial cells. So that suggests to us perhaps the anti-tumor function tumor-associated macrophage might not really target cancer cells themselves, but target this sort of critical cancer cell hallmark, which is this antigenic switch, sort of vasculature incorporation that is essential to promote tumor progression. Indeed, with this translocation, we saw a sort of a vasculature phenotype, or in other words, that in contrast to have more of this fragmented sprawling angiogenesis in the control tumor, as you would expect, in this TSC deficient macrophage mice, the vasculature become more organized, and that results in the induction of hypoxia response in the region that distant to the vasculature, and ultimately result in the cancer cell death, in this case, cleavable caspase-3 staining in the even more distant regions from the vasculature, sort of quantified here. So that suggests to us this anti-cancer mechanism is really targeting the cancer environment instead of cancer cell, in this case, sort of promoting remodeling of the tumor vasculature. The question is that how this reprogramming might be mediated. We followed the early publication, which to review that even for the endothelial compartment, there's a heterogeneity in terms of different types of endothelial cells. Some of them will have progenitor stem cell-like activity that can repopulate the new vasculature growth, versus others would be more differentiated cell. So in fact, one of the markers that defined is more of a progenitor-like endothelial cell in the mammary gland is the one that expresses high levels of this marker protein called protein C receptor PROCA. What we found was that, in fact, association with the vascular reorganization, with close association with tumor-associated macrophage within the endothelial compartment, is that we saw induced apoptosis or depletion of this PROCA-positive endothelial progenitor cells. For instance, in this F80 stands for tumor-associated macrophages. PROCA stands for this endothelium progenitor type of population. Cleavable caspase-3 again depict the cancer cell death. So as you can see, in the vicinity of this tumor-associated macrophages, there are many of this endothelial progenitor cells. They undergo cell death. Now this cell that's induced in close proximity to tumor-associated macrophages remind us of phenomena that has been more studied in developmental biology, which is so-called cell competition. So cell competition describes a phenomena in which the cell phase, or essentially whether the cell will survive or not, is really dependent on the relative fitness of these cells to their neighbors. Or in other words, that if, let's say, if you have all the cells in homozygosity, despite they may have relatively low fitness, they can still populate and survive, even, fulfill its normal function, tissue function. But if you make a mosaic system, for example, has this relatively less fit cells in the context of better fit cells, they will be actively eliminated, right? You can also have the cells have even high fitness, so-called super competitive cells, even the wild-type cells. In this case, if you make a mosaic, then the mosaic cells, a system in the tissue, even the wild-types can be eliminated. So it's really a function that involved to check the relative fitness and really to select for the best fit cell in the tissue. These observations were first made in Drosophila, but later studies to show this can also occur in mammalian species. For example, during early embryonic development, the epiblast differentiation were very much subject to this type of cell competition regulation. Perhaps it makes sense because you want to select the best stem cells to populate the whole organism as an individual. Now, what's interesting was that over the years, many of the molecular regulators of the cell competition has been reviewed. For instance, for the cell to become a winner cell state, high expression of MYC as a transcription factor and high activity TOC1 is typically associated as a winner state. And on the other hand, for example, loss of function of the tumor suppressor p53 can also give cell more of a winner cell state. But consider mTOC1 high activation can be associated with winner state, and the fact that TSC deficient tumor-associated macrophages express high levels of, have high mTOC1 activation, we ask the question, could the cell competition likely account for the sort of killing a depletion phenotype of endothelial cells? So we test this by doing an in vitro culture experiment, and indeed we could show that the co-culturing endothelial cells with tumor-associated macrophages, the TSC deficient macrophages can result in more of a selective killing or depletion of the PROCA-positive endothelial progenitor cells, much more so than the PROCA-negative endothelial cells, and also much more so than wild-type cells. That's provide a potential mechanism for how this tumor vasculature reprogramming can be suppressed by this sort of relocalized reprogrammed tumor-associated macrophages. So that's a story we just recently published, I think just probably 2 weeks ago, in Immunity. So in which we'll propose a model in which that So these tumor-associated macrophages can be reprogrammed to the point to have this more innate anti-cancer mechanism involve competition between macrophages and this tumor-associated vasculature to the point it would suppress this aberrant angiogenesis, which is essential for cancer to progress in terms of continuous tumor growth. And we think this again defines a unique mechanism how these macrophages can engage this unique type of innate anti-tumor response, more targeting tumor tissue as anti-tumor mechanism. Okay, so for the second story I want to share with you today, it's also again a story we followed up with this cell competition idea, and considering much of this sort of regulated cell competition are essentially genes typically associated with cell transformation. For instance, MYC overexpression, as we know, the MYC amplification associated with many malignancies. mTORC1, that's one we just discussed about. Loss of p53, that's another one. And in fact, cell competition has been speculated to be also critical mechanism to drive cancer cell evolution, right? Although, this has not been really extensively studied. So one of the study implied that this may take place in human malignancy associated with profiling c-MYC expression in various tumors, including breast cancer, but also many other cancer types. What was observed in human tissues is that high MYC expressing cancer cells typically associated with dying cell in the vicinity of that cancer cell. This can be other kind of MYC low cancer cells, but it can also be other stromal cell populations. Now, so that suggests in competition type of mechanism associated with MYC high expression may take place in malignancy, in cancer. But MYC is also a complex transcription factor because overexpression of MYC alone would not sufficient drive transformation. In fact, in many cases, it can even trigger cell apoptosis. Or in other words, the oncogenic function MYC has to be coupled with other regulators, right? So we're interested in this broad question and ask the question, could the MYC be potentially result in some cancer cell competitive phenomena? And then if that's the case, how potential microenvironment signals, including immune cell signals, might be modulated cell competition that takes place associated with gaining the some more window or aggressive cancer cell. So to this end, we analyze in this case human breast cancer database, and we found that the combination of MYC amplification and also this PI3K gain function mutation can really give the most aggressive human breast cancer that support some percentage of MYC with other oncogenic parts to drive more aggressive disease. So that sort of is interesting to us because we know the polyamido T model can already model to a good extent the PI3K gain function mutation because that technically activate the PI3K pathway. So we asked the question, could additional overexpression of MYC will make the POIMT tumor to be more aggressive, and whether we can start to see the cell competition phenomena. So we crossed this POIMT mice to a cree line in which MYC can be selectively expressed in cancer cells. Indeed, we see a really aggressive cancer cell phenotype compared to control POIMT mice. Now, what's also important was that this just in support of mTORC1 to be a critical regulator, also for the cell transformation or cancer cell phenotype, if we deplete mTOR, that can suppress tumor progression. You may say the suppression in the MYC POIMT model not as striking, but in fact, what we later, if you look into the cells that grow out of this sort of triple genetic transgenic mice, in fact, these are the escape mutants which have MYC overexpression, but mTOR still maintained. So this surely suggests that mTORC1 is a critical driver in terms of this aggressive tumor phenotype. So then we took a closer look in the tumor tissue, what happened in terms of this MYC overexpression POIMT cancer cells. So POIMT alone, as I showed you previously, they can already drive cell transformation. So it's a slow tumor growth, but also interesting is not associated just in the control POIMT tumor, much of a cell death. The MYC POIMT tumor is a much aggressive tumor growth, but it also is associated with much enhanced cancer cell death. But what's interesting was that if you look at what the dying cells are localized, in fact, they are all localized in the vicinity where all these high MYC expressing cancer cells. So the cells that dying are, in fact, all MYC low cancer cells. What's also interesting was that this high MYC cancer cells also associated with high mTORC1 signaling. This is very much consistent with the idea TORC1 is essential to support the aggressive tumor phenotype. So in the absence of mTORC1, in fact, if we look at the early tumor tissues, we saw a very interesting phenotype in terms of, in this case, in fact, MYC high cancer cells, they're more likely to die, defined by cleaved caspase-3 staining. Or in other words, that high mTORC1 activation is essential to enable MYC high cell to be a winner cell, right? So this TORC1 signaling is really critical for that to, for this sort of cell competition to take place. So considering this phenomena, we wanted to understand how this may be regulated. So in this case, we looked for differentially expressed genes. First of all, in humans with a subset that has both MYC amplification and PI3K mutation with the others. And then in the mice, we can just have MYC PRMT tumor with a control PRMT tumor. In fact, we have many pathways that appears to be associated in both cases, to be enriched in both cases, right? Among the ones that really enriched in the MYC PRMT or MYC PI3K mice are the ones really associated with various enhanced metabolism, including amino acid metabolism, but as well the cellular response to starvation. That really suggests that this sort of MYC high, PI3K high type of tumors that really highly metabolically active and also very demanding in terms of nutrient acquisition because associated with cellular starvation. So these we asked the question, could these tumors be sensitive to the variance in terms of the amino acid that can be acquired from the diet? So we used, in this case, the low protein diet and asked the question, would that affect tumor progression? We're interested indeed, the low protein diet can really suppress progression of this MYC PRMT tumor specifically, not in fact affect that much the slow growing PRMT tumor. So we thought initially this just can be a simple mechanism because I showed you or implied previously mTORC1 signaling, as I showed you in the previous slide, is enables this winner state. And mTORC1 activation, in fact, dependent on nutrient acquisition or specific amino acid acquisition. I thought this could just be a simple mechanism, how amino acid needed to support high TORC1 activity in cancer cells as sort of cell autonomous mechanism. This is until we looked into in the tissue how the low protein diet affect mTORC1 activation. So first of all, associated with reduced tumor growth, indeed, on the low protein diet condition, this epicam positive cancer cell that will have low mTORC1 signaling compared to normal protein diet condition. But what's interesting was that under low protein diet condition, not all the cells TORC1 signal is inhibited. There's still mTORC1 high cells in the tumor tissue microenvironment. They are not epicam positive cancer cells. There must be other kind of stromal cell population. They turn out to be macrophages. It's F40 against the macrophage marker. So it's curiously, in fact, I showed you previously, right, in PRMT model, there's really abundant macrophage in the tumor parenchyma, right? But the MYC PRMT tumor, in fact, that depletes macrophages as well, right? Not only can trigger death of neighboring low MYC cancer cells, even MYC PRMT, even the macrophages can just be depleted in a MYC PRMT tumor. But under normal protein diet condition, but they can come back under low protein diet condition, but in a way that it will have high mTORC1 signaling. So, or in other words, as a low protein diet will just reprogram tumor-associated macrophage response in a MYC PRMT tumor so that can repopulate the tumor tissue environment and in a sort of mTORC1 high state. It's sort of quantified in terms of mTORC1 activation in macrophage, and it's sort of associated with also big TAM cell size compared to what you see in the PRMT tumors. I also just keep in mind, in this case, all wild-type, genetically wild-type macrophages, right? The reprogramming is triggered by the diet on this specific genetic background. With this phenomenon, ask the question, could this reprogrammed macrophage might contribute to the tumor phenotype? So we could test this by just depleting macrophage with anti-CSF1 receptor antibody. So that depletes the macrophage, and really exciting was that in these cases, tumor suppression phenotype is reversed. This is associated with also gaining more TOC1 signaling in epicam-positive cancer cells. Or in other words, this low protein diet triggered suppression TOC1 signaling and tumor growth is not cancer cell autonomous. It is all dependent on this macrophage reprogramming instead of simply because of its act on the cancer cells directly. So that's really intrigued us. Then we really asked the question, first of all, will this high mTOR1 activity in macrophages be essential for this sort of reprogramming and the tumor suppression? So we could, in this case, delete tumor mTOR1 in a macrophage with a specific query, and also the bone marrow chimerical approach to ask the question, if we delete mTOR1 in macrophages, will affect the response in the tumor? And that turned out to be the case. Because if we delete tumor-associated macrophages, this reprogrammed macrophage response will be reversed. We're not going to see this big F480 mTOR1 high macrophage again, because the mTOR1 knockout macrophage will not have mTOR. In the absence of mTOR, this reprogramming, repopulation macrophage in the tumor microenvironment is suppressed. Macrophage is no longer there. And associated with depletion macrophage, again, as I showed in the previous macrophage depletion studies, now this mTOR1 signaling shifted from over macrophage dominance to cancer cell dominance, and this tumor growth phenotype is reversed. So in other words, that is, so essentially mTOR1 signaling tumor-associated macrophage is essential driver of this macrophage reprogramming. Then the question is that how can a low protein diet result in enhanced macrophage mTOR1 signaling? Because it's counterintuitive. We typically think you need high amino acid to drive mTOR1 signaling. Now we have this sort of counterintuitive opposing phenotype. The low protein diet condition can, in fact, paradoxically result in macrophage TOC1 activation. So that's really intrigued us. To this end, we looked for what might be defective with this low protein diet in terms of specific amino acid concentration in the tumor interstitial region. So we did an amino acid profiling experiment to the point we could show, in fact, there's only a few amino acids concentration is changed with the low protein diet. And this involves a critical amino acid called the leucine. Now, leucine is interesting to us because leucine is no amino acid that can be involved in regulating this RAG-dependent nutrient sensing pathway. Again, this is more of an introduction of the RAG-GTPase-dependent nutrient sensing pathway. That is, small GTPase is associated with recruitment and activation of various type of so-called effectors of small GTPase, right? So one of the effectors of small GTPase is mTOR1 itself. That is why how typically most people will what they think, with amino acid abundance, they can promote mTOR1 activation, because with amino acid can suppress this another so-called GATA1 complex, which is a suppressor of the RAG-AB small GTP in its more active state. And this RAG-AB in the GTP state is going to recruit TOC1 to activate mTOR. So that's why, with abundant amino acid, these parts can be activated. Now, yes, still with this RAG-GTPase, there's another effector that can be regulated in response to sensing cytosolic amino acid. Is this TFEB-E3 family of transcription factors. So this family of transcription factor will be sequestered on the surface of the lysosome if there's abundant amino acid in the cytosol. But in the absence of amino acid, so TOC1 will not be recruited to the RAG small GTPase, and there's another complex called folliculin that really regulate this RAG-CD in the GDP state. But with the flipping of the RAG-CD to the GDP state, not the TFEB can translocate to the nucleus to induce a class of target genes. Many of them are involved in the engulfment process. You can view this as a sort of backup strategy, how the cell can activate this engulfment-dependent nutrient sensing pathway, can try to correct this starvation phenotype that's sensing the cytosol, right? So the sort of cells will adapt to low amino acid, try to find other ways of nutrient acquisition. Now, considering the phenotype we see, because we see the starvation, we see even high TOC1 activation in macrophages, we thought, could this potentially develop an alternative TOC1 activation pathway, perhaps depending on this nutrient acquisition through phagocytosis, not so very much cytosolic amino acid acquisition through transport-dependent mechanisms that can potentially activate an alternative TOC1 pathway that can even be more robust than the RAC TTPS-dependent TOC1 signaling pathway. That could potentially explain why, under low protein diet condition, mTORC1 to be even more active in macrophages. With the assumption macrophages are phagocytes, they have even more robust activity for engulfment. So that's sort of the model we try to test to explain the phenomenon. Indeed, in line with that particular model, we could show TFEB3 is active by tumor-associated macrophages. This is nuclear translocation TFEB3 only under low protein diet condition in the mixed pure empty tumor, not in the pure empty tumor. So are they really important? The TFEB3 transcription factors, again, we did the same targeting, deleting this transcription factor in tumor-associated macrophages. That indeed also completely reverse the reprogramming. Macrophage will not be populate the tumor anymore, and TOC1 will not be activated, and now the TOC1 signal will switch from macrophage to cancer cells, and tumor progression is suppressed, right? Now, that really mediated through this sort of amino acid cytological sensing RAC GTPS pathway. We can test this because much of the amino acid starvation sensing is by the GATA1 complex. So we could also delete the GATA1, ask the question, in the absence of GATA1, TFEB2 will not be able to translate to the nucleus. Would also drive all this reverse the type of reprogramming that we just we just discussed about. So it's the same trick of the genetic manipulation of the GATA1 complex in the macrophages, and that indeed is essentially, as I showed you previously, the TFEB3 translocation will be abolished if you don't have GATA1 complex. Macrophage reprogramming is abolished, and the TOC1 signal switched from macrophage to cancer cells, and tumor phenotype is suppressed. Now, if let's say what really going on is about TFEB3 dependent nutrient acquisition and TOC1 activation in macrophages, if that's really the prediction, we could, if that is the model, we could predict if we depleted follicular or even RAC GTPases themselves, because that would result in consistent TFEB3 nuclear localization, right? So if this is the case, can we really even suppress tumor growth under normal protein diet condition? Because now we can ectopically activate this pathway in tumor-associated macrophages, right? So that appears to be the case. Even under normal protein diet condition, if we delete follicular or the RAC family small GTPase, we can trigger the de novo macrophage reprogram even without the diet change, and resulting in the expansion of this TOC1 high macrophage in the tumor microenvironment. In fact, that can suppress TOC1 signaling in cancer cells. That results also in reduced tumor progression. Or in other words, that this nutrient acquisition appears to be very much competitive between tumor-associated macro and cancer cells. And in this case, if you even have this, appears to be, mTORC1 high macrophage to be triggered by this sort of manipulate the nutrient sensing pathway around TFEB3 regulation, we can gain this unique phenotype. So then the question is, what is really mediating this reprogramming, or indeed whether or not this engulfment pathway different TOC1 activation indeed takes place in this context, because we propose an alternative pathway on TOC1 activation. So that's sort of the question. We think we thought this might be the case because the observation we made was that this reprogrammed macrophage with low protein diet in the mixed pure empty tumor, there's really highly phagocytic in this case, defined by engulfment, a lot of tunnel-positive apoptotic nuclei from dying cells. We further show, in fact, this engulfment indeed are these dying cancer cells, because we can label the nuclear cancer cells with H2B, the histone 2B and cherry, a fluorescent marker. We can also mark just macrophage nuclei with H2B GFP, in which we can show, in fact, in this tumor-associated macrophage, on the low protein diet condition, the abundant this M cherry positive nuclei, which will be the nuclei from dying apoptotic cancer cells. So this macrophage is really actively engulfing a lot of dying cancer cells in this case. To really test whether this engulfment pathway is critical for the reprogramming, so we focus on this engulfment regulator, the PIK5 kinase, which in fact is essential to promote phagosome and the endosome maturation. In fact, it's essential for not only for phagocytosis, but also macropinocytosis, which can take place in cancer cells for its ultimate sort of nutrient acquisition and that's, define some of the engulfment pathway of a nutrient acquisition. So we could show, in fact, if we have the macrophage deficient for PIK5, that can result in diminished engulfment of apoptotic cancer cells with the engulfment assay just introduced. So that is also, in fact, if you don't have PIK5, very much similar to what I showed without TFEB3 or without mTORC1, that can result in defective reprogramming these tumor-associated macrophages. Again, the tumor growth phenotype, suppression phenotype is reversed. Now, if indeed what's really going on here is about macrophage cancer cell competition for nutrient through engulfment, then we could ask the question, if we now make the cancer cell not be able to engulf nutrients through macropinocytosis, again, we can enable that by depleting PIK5 in cancer cells, right? So then we did this mixed matched bone marrow reconstitution system, which we could show that this gained tumor growth phenotype with abolishment of this tumor-associated macrophage response to PIK5 deficiency will not longer take place if the PIK5 is deleted in cancer cells. Or in other words, that the cancer cell engulfment activity, so PIK5 dependent activity, is essential for how this sort of macro depletion triggered gaining of TORC1, gaining of tumor growth. This kind of also reflect in this particular standing in which we could show only if the PIK5 is sufficient in cancer cells, we can gain this high TORC1 signaling in cancer cells. Or in other words, even for the mTORC1 to be highly active in cancer cells, they need to depend on the nutrient acquisition pathway, right? Then the question is, can we really directly show this is about competition for engulfing dependent nutrient acquisition? So we could test this essentially by feeding the tumor tissue either with apoptotic cell, which we can label with one of the fluorescent marker, or apoptotic bodies, that's smaller particles that from disintegrated apoptotic cells, right? Now, consistent in line with the idea, macrophage phagocytes, they're really sort of professional eaters. They can just engulf materials in a much efficient manner. So, in fact, macrophages can effectively engulf apoptotic cells as well as apoptotic bodies in with a low protein diet. It's essentially with wild-type tumor-associated macrophages. And these are very much dependent on PIK5, because if we don't have PIK5, this engulfment is very much diminished, as I showed in the previous slide. Much more of the apoptotic cell, but to also a large extent apoptotic body. Now, what's really exciting was that in this case, in the absence of the engulfment activity of macrophage for this sort of engulfment activity, but the cancer cells can gain this engulfment activity, but curiously, specifically just for apoptotic body, not for cancer apoptotic cell, right? So this sort of quantification over here. So that sort of will come back to the model to explain this particular. Now we consider another layer of cell competition-based anti-tumor immune response involve macrophage reprogramming. Essentially, this specifically the MYC-POMT type of a tumor. So that these tumors, cancers that can be really, really high metabolic active. They really need dependent, in fact, in this case, local engulfment to acquire nutrient from this dying, less fit cancer cells, more specific apoptotic bodies, not entire apoptotic cells, to further fuel high TORC1 signaling, and that's the sort of winner state of better cancer, aggressive sort of tumor cancer cell state. Now, what's curious was that these particular competition between cancer cells can be really disrupted if we reprogram this tumor-associated macrophage response, either with a diet, just simple put the mice with a low protein diet, or genetically, even on the normal protein diet, but deleting follicular rack GTPases, that resulting in this activation TFEB3 transcription factor, and thus resulting in this really robust engulfing different nutrient acquisition, and it's still yet poorly defined, still remain to be defined, this alternative mTORC1 activation pathway, and that really sort of outcompete this even high MYC cancer cells for nutrient acquisition, and thus all these cancer cells, even high expression of MYC, they will undergo apoptosis. So that's sort of the model we have to express this phenomenon. Put these both stories together, hopefully we can just really show you this sort of interesting cell competition-based cross-regulation, and curious all involve sort of macrophage TORC1 signaling pathway, and also one involves in the P1 model itself competition with endothelial cells, the other in fact in the MYC P1 tumor competition between macrophages and cancer cells. Now, since it's, this probably for this audience more interesting for osteosarcoma, I just want to emphasize that although, what we showed is more in the breast cancer model, the general concept can also be applied to osteosarcoma. In fact, for instance, VEGF-associated tumor angiogenesis is also, sort of a nice summary of molecular targeting in osteosarcoma in a recent review. Think about the pathway maybe target osteosarcoma. Like VEGF, in fact, itself is considered how, in terms of, supporting a vascular environment in osteosarcoma, like in many other cancer types. So this, in this sense, we can wonder maybe this macrophage compartment can also be reprogrammed to also achieve similar kind of anti-angiogenic effect. But also curiously, the MYC and the TOR is very much highly induced in many of the aggressive osteosarcomas, right? So we'll be really curious to know, for example, whether this particular type of macrophage-dependent cell competition phenomena can also be reprogrammed as a way for therapy osteosarcoma. I think if the patients are curious, you might even entertain the idea, potentially, change up the diet can already be a way to do this. But obviously, we'll be interested to see whether you can also find other ways to manipulate even genetically tumor-associated macrophages as a way even for cancer therapy without have to change the diet. I would be curious, if the foundation is interested in this topic, although, we could potentially think about developing osteosarcoma models to test all these type of ideas. But, if that's an interest, they will be interested to develop the model to further explore this for osteosarcoma. With that, I'll just acknowledge, also, as I said, it's typically not think about, like macrophage control the vasculature, but certainly that can be relevant as well, MYC and TOR. So maybe I just save some time for discussion. I will not sort of go through in detail the acknowledgement slides, but I'd be happy to take any questions for discussion.
Thank you so much, Dr. Li. And please, anyone, if you have any questions, feel free to put them in the chat so that we can, in the Q&A, so that we can ask Dr. Li. But you kind of touched on this with this slide, because I was just curious, this was studied in a breast cancer model. And so, as you noted, MYC is overexpressed in some kind of aggressive osteosarcomas. And so, is it transferable, the MYC kind of PI3K signaling, and it was the basically the RAG GTPase that inactivates the TFEB and TFE3, right? Mm-hmm. Is that pathway mechanism, is that transferable to other cancers, or is MYC, can it act differently in different cancers? And I guess, and also has this been studied in other types of cancer models besides breast cancer? Validate that.
Yeah. We have other models more involve MYC-TOR cooperation. That really appears to be a general synergistic pathway. We saw, in fact, we have a new model of peripheral T-cell lymphoma, just drive by with the synergy. So we would think that this sort of molecular cooperation to support the winner cell state with the cooperative TOR and MYC, it will be a general phenomenon. That's also occurs in healthy cell, like a lot of healthy competition in developmental processes, all sort of the similar media by this two pathway. I would think this will be a broadly applicable different cancer indications. And I think it will probably still remain to be determined to what extent whether this macro reprogramming with low protein diet also occur in these other conditions, but we have looked into preliminary some other model appears to be the case. Osteosarcoma could be, essentially be another case worth looking into. Because I looked briefly into the field because I have not thought so much about osteosarcoma yet, but like MYC at all is like very highly pathway with activate TOR is very prevalent. So is also MYC amplification. So I would think this can be worth considering for osteosarcoma.
And so I don't know if you saw, there was this paper that came out, I think just this month, and I think it was from the Netherlands Cancer Institute. And so their paper, did you see this? It was basically, it said MYC is a clinically significant driver of mTOR inhibitor resistance in breast cancer. And I was wondering if you could help me connect the dots on that. Because the other thing is like the simple read for, a layperson is like, oh, okay, like mTOR inhibition, right? Like how helps, decrease the signaling. And so, why doesn't an mTOR inhibitor work against this kind of MYC overexpression?
Yeah. So, I think that's a good question because, any kind mTOR is such a critical factor and usually depends on how extensive we can suppress TOR. And so therefore, it is possible like MYC can be escape mechanism for TOR inhibitor. Could just be, if you have because there's a mechanism how MYC can enhance TOR signaling. We have a story that, we're wrapping up to demonstrate that. So that we could imagine that we have high MYC activity, it can make the TOR inhibition to be more difficult to be achieved with the drug programs. Because these two parts cross-regulate. Like as I showed in problem on the slide, the MYC pure MT has much, much higher mTOR than pure MT alone. Despite you would think, pure MT itself will active PI3 kinase already activate TOR itself, right? But yes, the MYC transcription will really give a much higher level than mTOR. So in this case, if you consider without TOR inhibitor, probably difficult to suppress TOR to the level have a substantial tumor phenotype, you have MYC overexpression.
This was a lot to digest. Thank you so much. And thank you for all the helpful slides. And I think it sounds like you were generous enough to allow us to share these with our registrants so we can spend more time with them. But thank you so much. And we would love to put you in touch with some folks that are studying MYC and osteosarcoma to see how this might be possible.
Thank you for having me.
Yeah, thank you. So thank you so much for joining us and for making it better for cancer patients, Dr. Li. More information on this and all osteobites can be found on the MIB Agents YouTube channel, on our website at mibagents.org, and at your favorite podcast place. And please join us next week. We're going to be talking to Joshua Nash, who is a PhD candidate under Dr. Adam Schlein at the Hospital for Sick Children in Toronto, Canada. And Joshua's research focuses on leveraging transcriptomics and machine learning to understand the overarching biology of sarcomas and mesenchymal tumors and to develop more molecularly informed and prognosticating diagnostic subtypes. Josh is going to join us on Osteobites to discuss his work developing a multi-scale transcriptional atlas of sarcoma. You can find our Osteobites lineup for the next 2 months on our website. And if you have any ideas for future topics that you'd like to hear about, please share them with us at events at mibagents.org. Thank you again, Dr. Li, Walker, and for all of you for spending an hour with us today. We hope to see you back here on Osteobites next week on the 7th when we talk to Joshua Nash. Thanks, everyone.